by the time I finish rambling on, it'll be 5 past. You can take it away.
Sure.
So
okay,
then you don't, got plenty of time for your talk. So good afternoon, everyone. Morning in Stockholm, where Rachel is joining us. It's a great pleasure to host her Zoom seminar today, and great to see someone from my old lab where I did my postdoctoral training in the lab of Professor David Drew at Stockholm Uni. David's labs is a transporter heavy lab, and Rachel's going to be telling us today about the structure of a tripartite ADP independent periplasmic transporter involved in bacterial virulence. So she did her PhD at University of Canterbury, originally from Christchurch, and then did a postdoc there in the same group before moving to David's group in Stockholm. So really large welcome, Rachel. Really looking forward to your talk. Thanks so much again for taking the time to share your research with us. Take it away.
Cool. Thank you so much. I'll just share my screen. Nice. And everyone can see that okay, I assume? Cool. So thanks, Joe, for the introduction, and thank you very much for the opportunity to be able to present my research today in the Southern Hemisphere, although online. It's been a really long time and I've been really looking forward to it. So I'm going to present today, as Joe said, the structure and function of the tripartite ATP-independent periplasmic transporter, also known as the TRAP transporter. So I started this project actually around 8 years ago during my PhD in the group of Ren Dobson at the University of Canterbury in New Zealand. And then I continued on with this project along with other projects during my postdoctoral position there. And then I moved to Sweden approximately 2.5 years ago and joined the group of David Drew. Here I'm working with mammalian membrane transporters, but I've continued to dabble with this one on the side because it has been a really tough one to crack and it's really interesting. So that's what I'm going to share with you today. Okay, and just to give you some background for what I'm going to present today, we'll start with sialic acids. So sialic acids are really large, negatively charged sugars. They're a really large family there's over 50 structurally distinct types of sialic acid. And the most common sialic acid is shown over here on the right-hand side. And this is known as N-acetylneurominic acid, or you may have also heard of it as Nu5AC. And this sialic acid is ubiquitous across all organisms, and it is the most abundant sialic acid found in humans. And so the surfaces of eukaryotic cells are coated in these things called glycans or glycoproteins. And these glycans are comprised of a range of different sugar molecules. And in general, the terminal most sugar molecule on these glycans is usually a sialic acid. And these sialic acid coated glycans or glycoproteins are really highly abundant in mucus-rich environments such as the human gastrointestinal tract or the respiratory tract. And interestingly, these are also the same environments that many common cell and pathogenic bacteria like to colonize and persist in. And here, these common cell and pathogenic bacteria have adapted and evolved to be able to utilize these sialic acids, especially in environments where the glucose is really limited. So what they do is they scavenge it from the surfaces of our cells, and then they import it into their own cell via a sialic acid transporter over here. Once imported into their cell, they can either degrade it into a nutrient source, or they can use it to basically sialylate their own cell surface. So they put the sialic acid on their cells, and then essentially they then camouflage themselves from the host's innate immune response. And so during my PhD, I started working with the 5 enzymes involved in the sialic acid degradation pathway. And it was about halfway through this, I became really interested in how sialic acid is actually imported into the bacterial cell, and then started working on them then. So there are 4 different families of sialic acid transporter, very different families actually. And although there are some exceptions, most organisms generally only have one type of these sialic acid transporter. And I'm going to take you through these in a little bit of detail first. So first up, there's sialic acid transporters that belong to the major facilitator family, and these co-transport both sialic acid and protons across the bacterial membrane. Next up, there's the sialic acid transporters that belong to the sodium solute symporter family, and these co-transport both sodium and sialic acid across the membrane. And prior to the work I'm presenting today, this was the only structure that we had available for a sialic acid transporter belonging to the sodium solute symporter family. And it was one that we solved during my PhD and postdoc position at UC. And interestingly, I mean, there are other transporters structures available for this family, but for this sialic acid transporter in particular, we found that it had a new sodium site not yet seen in other family members of the sodium solute symporter family. And so next, there are sialic acid transporters that belong to the ATP binding cassette superfamily, or more commonly known as the ABC family. So these transporters are primary transporters. They use this secreted substrate binding protein up the top here to bind the sialic acid and deliver it to the membrane component for import. And they energize this import by the energy derived from ATP hydrolysis. And then lastly, there is the tripartite ATP-independent periplasmic transporter family, which I'll call TRAP transporters from now on throughout this talk, and this is the focus of this talk. And similar to ABC transporters, they also have this SIAP substrate binding protein to scavenge sialic acid and deliver it to the membrane component in a manner sort of analogous to the ABC transporters. But these transporters are not primary transporters, they're actually secondary transporters. So this makes them really exciting because they are the only secondary transporter we know of that uses this substrate binding protein like a primary transporter to deliver their substrate. And so instead of using ATP hydrolysis, they energize their transport using energy in the form of a sodium gradient to import their sialic acid. And out of all of these families in general, so the first 3, like we have some structures for, and maybe not sialic acid transporters, but other transporters in a range of different conformations. And in general, we kind of know mechanistically how they work. But for the TRAP family, there are no structures at all, and apart from the substrate binding protein, and we don't really understand mechanistically how they work, which is a shame because they're very interesting. And so there's plenty of literature out there that indicates that sialic acid is a key nutrient for bacterial pathogens to colonize and persist in environments. And the TRAP transporters have been specifically implicated in bacterial virulence and pathogenesis. And interestingly, TRAPs are widespread in bacteria and archaea, but they are not found in eukaryotes. So they're not found in mammals and humans. So this opens up a really nice avenue for developing new antimicrobials that are likely non-toxic to humans, and you can then target pathogens that use these TRAP transporters during infection. So the TRAP family is a really large family. It's not just limited to sialic acid transporters. There are many different TRAPs out there, and they import a range of different solutes, shown, for example, on the left-hand side over here. So these solutes are generally organic acids or other similar molecules, and they generally contain a carboxylate group or a sulfonate group. And so TRAP transporters are really specific for a particular ligand, and this can be attributed to the high specificity of the substrate binding protein up the top here. So we focus on the sialic acid TRAP transporter system, which is called SIAP-QM. SIAP describes the substrate binding protein, SIAP, and SIAQM describes the transporter protein, which is comprised of 2 different subunits, the Q subunit and the M subunit. And so we had 2 main research questions because we don't really know so much about these proteins, but one was, what is the structure of the TRAP membrane transporter? So As I mentioned, there are no structures of the transporter component, but there are plenty for the substrate binding proteins. And then our second question was, how does the substrate binding protein interact with and deliver substrate to the transporter? Cool. And so, like being membrane proteins, these trap transporters are really difficult proteins to work with in the lab. We spent many years screening various different constructs for protein expression and screening them for their stability during purification, and also for their stability in any future experiments we were doing or wanting to do. We also spent many years crystallizing these proteins. We could get beautiful crystals for some of them, and they would diffract to 6 or 8 angstroms, but they could never be phased. And we also couldn't improve the diffraction beyond that. And of course, we also did many cryo-EM trials, but at that stage, the trap transporter is considered quite small for cryo-EM, being only 70 kilodaltons. So at this stage, it wasn't successful. But finally, we were able to solve the first high-resolution structure of a trap transporter by cryo-EM. And we solved it, the SiaQM, sialic acid transporter, from Photobacterium profundum to 2.97 angstroms. And we did this in complex with an engineered megabody. As I said, the trap transporter is relatively small at 70 kilodaltons for cryo-EM. So this megabody enlarged the size and helped us with particle alignment and orientation during the data collection and data processing. So trap transporters, as I mentioned earlier, are made up of these 2 different subunits. First is the Q subunit, also known as SiaQ, and second is the M subunit, also known as SiaM here. And so our megabody here was placed, was located or bound at the periplasmic face of the transporter, so at the top of the transporter up here, and it makes contacts with both the Q subunit and the M subunit. And you might notice that the megabody here looks relatively small for 55 kilodaltons, and that's actually because we couldn't see the entire megabody in the cryo-EM maps. We can actually only see the nanobody portion of it. Oh, and I should also mention that we solved this structure in amphipole, so it wasn't in detergent. We purified in detergent and then we exchanged into the amphipole for the cryo-EM. We found that this was a little bit less noisy, and it was really, the protein was really happy and stable in the amphipole. And so a megabody is made by, you first have a nanobody and then you engineer a scaffold protein to bind to it, which is the large part of it. And in our representative 2D class over here, you can see very nice transmembrane helices. And then we can also see the nanobody here, which is quite rigid. And then the scaffold protein is actually just this big blur up here. So this tells us that it was quite flexible and probably just flopping around there. And that's why we can only see the nanobody in our cryo-EM maps. So I'll take you through the data collection and processing very briefly. So we collected using a Titan Krios with a K3 detector, and this was done at SciLife Lab in Stockholm. We collected almost 11,000 images, and it's relatively small, but over the left-hand side here, you can see a representative micrograph, and the particles are really nicely distributed here. So from our 11,000 images, we got almost 6 million particles. And then we used these and did a whole bunch of 2D classifications. And you can see in these 2D classes the blur of the megabody as well, which is quite cool. And you can even see the transmembrane helices. And so after cleaning up the 2D classes a little bit, we were left with about 1 million particles. And then we cleaned this up even further by doing an initial reconstruction or an ab initio reconstruction. And at the end here, we were left with about 600,000 particles to work with. So after many rounds of refinement, in the end, we were left with gold standard maps with a global resolution of around 2.97 angstroms. And if you have a look at the bottom right here at the local resolution map, you'll see that the local resolution actually extends to even 2.2 angstroms in some areas. And this is indicated in this blue and cyan color, predominantly through the middle here. And then the lower resolution is sort of at the top of the nanobody here, which makes sense considering we couldn't see our megabody in the map. And here we have some representative EM density just to show you how well we could build our model into our cryo-EM maps. So at this resolution, the side chain positions are really clearly resolved throughout most of the structure. All of the transmembrane helices and majority of the extra and intracellular loops could be really accurately modeled, which was really nice. And solving our structure of a trap transporter allowed us to define traps as elevator type transporters. And so an elevator type transporter is comprised of two separate domains. So first we have this fixed barrier domain, or it's also called a rigid scaffold. And this is sort of fixed in the membrane and it doesn't move almost whatsoever. And then there's the transport domain in orange here. And this is the domain that's likened to an elevator. So this moves up and down in the membrane against this fixed barrier domain. And this is a really large and really dramatic conformational change for a transporter because it has to move right through the membrane. In comparison to other transporters that like I'm used to working with, they generally just open and close on either side of the membrane to allow alternating access. And they do this by like small local gating rearrangements. And of course, elevator transporters also have these small local gating rearrangements. So there's a gate here indicated in the gray, and this opens and closes to allow obviously the substrate to bind when the elevator's at the top, and then opens again for the substrate to be released when the elevator is moved to the bottom. And so we solved our structure, which is shown on the right-hand side over here, in the inward facing and elevator down position. We have this really large solvent accessible cavity at the bottom here, which is open at the inside, and this is where the sialic acid would have been bound. And so, as I mentioned, trap transporters are comprised of two domains, the Q domain and the M domain, which you can see here and here. The Q is the scaffold and the M is the transport. We have the Q subunit, which is made up of 4 transmembrane helices, and the M subunit is made up of 12 transmembrane helices. And this M subunit has 2 inverted repeats. One of them is here, and the other one is here. And these inverted repeats are also comprised of, so they're made up of a scaffold domain and a transport domain. And this M subunit actually shares, although it shares no sequence homology with any other transporters, because trap transporters have almost no sequence homology to anything else, they do share a similar fold and topology to other transporters. And I'll talk about them in a little bit more detail later. But these other transporters that they share their fold and topology with are also elevator transporters, confirming that we also, for traps, we do have an elevator transporter. But the scaffold domain over here, which forms interactions with the scaffold domain in the M subunit, is a new feature for elevator transporters. So we only see this in the trap transporters, and it seems to extend the scaffold to make it even larger than what we've seen before. So if we take a look at the interface between this Q subunit and the M subunit, so the Q subunit on the left interacts with the scaffold domain of the M subunit. So as I said, the M subunit has also both a scaffold and a transport domain. And this interaction between these two subunits is, it bears a large surface area of around 2,400 angstroms squared. And this interaction is, is dominated by hydrophobic interactions and stacking interactions, making it a really, really tight interface. And we also have these 4 residues: arginine 47, arginine 32, glutamate 237 and aspartate 50, which form these salt bridges to help bind these two subunits together. And something else I'd also like to mention is that in some organisms that have transporters, this Q subunit and M subunit is co-expressed as two separate polypeptides that come together in the membrane. And in other organisms, they are expressed as one polypeptide that is already tethered together. And so for the system that we've been working on, the SciQM from Photobacterium profundum, this is a non-fused system. So these are actually two separate proteins that do come together and form this really tight interface that you can see here. So although we had sialic acid in our purification conditions and our blotting conditions, unfortunately, we don't see the sialic acid in our cryo-EM structure, but we do see this sialic acid binding cavity here that it would likely bind in, and this is in between the two inverted repeats. And it's also nestled in between these two broken helices. So one is here and the other one is here. And in between these two broken helices, we seem to have these sodium sites. So there's one sodium site here and one shown here. We have very weak density for the sodium in the EM maps, but in both of these sites, we have a really nice coordination pattern, which is consistent with binding sodium in these areas. And also both the sialic acid site and both of these sodium sites are really highly conserved in trap transporters, supporting that we do in fact have sodium bound there. And also we did a structural comparison with a related transporter. It's not related in sequence, but it is related in its fold and topology, as I mentioned earlier. And this is called VC-INDI, and this supports our structural assignment of the sodium sites. So on the left-hand side over here, we have the sodium sites in SciQM. We have Na1 and we have Na2 over here in between these two broken helices. And in the middle here, we have VC-INDI, sodium 1 and sodium 2, also with sodium sites nestled between these two broken helices. And in this structure, they also had their substrate binding site, further confirming that we have our substrate binding site here in between these two sodiums. And then when we overlay these sites, you'll see that they're in a relatively similar position. And so this supports our assignment further of the sodiums that we see. Cool. So the structure that I've been talking to you about, as I mentioned, was solved using Amphipol. And we recently solved another structure of SciQM, the SciQM trap transporter from Photobacterium profundum, but this time we solved it in nanodiscs. And this showed us that lipids are likely very important for function. So we solved the structure to a similar resolution of about 3 angstroms. And over here in red here, we see some lipid density bound at the scaffold domain of the membrane transporter. And at the top of the transporter here, which we see in both structures, we have these 3 tryptophan residues. And tryptophan residues are known to sort of anchor a membrane protein in the membrane. So we think that the lipid at the scaffold domain here, which needs to remain really rigid in the membrane, and these tryptophans are contributing to sort of anchoring this in the membrane and contributing to its rigidity. Around the backside over here, we have some more lipid density. So this is 120 degrees around the other side. These lipids bind to the M transport domain, which is just next to the scaffold domain over here. And this lipid density is also in an area where there's a small patch of positively charged residues. So this indicates to us that the lipid binding here is binding with a negative charge. And we think that, so we hypothesize that because we have this large elevator movement that we need, we have really specific lipids binding at specific places and making specific interactions around our transporter. So the first, of course, was the lipid to help anchor the transporter in the membrane, and the second one is this lipid near at the transport domain. And here we think that this is assisting with the elevator movement. So it's allowing this large sliding motion through the membrane and essentially sort of greasing the elevator for its transitions that it needs to take. And so, as I mentioned, the trap transporters have an extended scaffold relative to other elevator type transporters. And this, we think, enables trap transporters to operate as monomers, as opposed to the typical oligomers that you see for other elevators, which are usually, majority of the time, they're usually dimers. So to test this, we did analytical ultracentrifugation experiments. And in an analytical ultracentrifuge, this will give you, you monitor the particle size and shape in solution as it sediments over time. And from this, you can get a sedimentation coefficient for the species that are present in the sample. So here we see the sedimentation coefficient for the megabody. In green here, we see psi-QM, and in orange here, we see psi-QM plus the megabody. And from this data here, we can then calculate a frictional ratio, and a frictional ratio kind of describes how spherical or how round a particle is in solution. And so the closer that value is to 1, the more spherical it is, the higher it is, the more elongated the particle is. And so for psi-QM by itself in green, we get a frictional ratio of about 1.2, and this corresponds to a protein mass of 70 kilodaltons, which is almost bang on what our trap transporter is. It is 70 kilodaltons. And if it was a dimer, we would have expected to see a frictional ratio up at just above 1.6. So these experiments did indeed demonstrate that our Photobacterium profundum psi-QM system that we've been working with is a monomer. And this is also consistent with what we saw on the cryo-EM grid, so, because our structure was also solved as a monomer. And it is likely this scaffold that enables this to happen, because other elevators don't have this. And so our collaborators at the University of Calabria in Italy did some proteoliposome substrate uptake assays for us. And so we sent them protein, and they did this by reconstituting the protein on the left over here into liposomes in the presence of valinomycin. And this valinomycin selectively pumps out potassium from the proteoliposome, and this creates a membrane potential. And so we find that the uptake of sialic acid occurs in the presence of an inwardly directed sodium gradient. So in green here, you can see the substrate uptake over time in the presence of both sodium and a membrane potential. And in orange, we have uptake in the presence of the sodium. So you can see with the membrane potential that we get enhanced transport. And down the bottom in purple down here, we have no sodium present, and also some other controls that were done. You can see you get no substrate uptake without the sodium gradient. We also found that the uptake of sialic acid occurs only in the presence of the CIP, substrate binding protein. So without CIP, at 0 micromolar here, we have basically no transport, and transport increases once the CIP concentration is increasing. So it's concentration dependent. And again, you can see that transport is really enhanced in the presence of this membrane potential here. And so consistent with our structure and the sodiums that we see in our structure, and also consistent with other related structures such as the VC India that I talked about earlier, we find that transport is electrogenic. We get a Hill coefficient of 2.7, meaning that sodium and sialic acid transport is cooperative. And being electrogenic means that it carries a net charge across the proteoliposome. And because sialic acid already has a negative charge, it's going to need more than one positive charge to be able to transport. So we see that at least 2 sodium ions are being co-transported for every sialic acid molecule during each transport cycle. And so despite extensive trials in detergent, in amphipole, and in nanodiscs, complex formation is difficult or basically impossible to capture for us by cryo-EM. And we think that this is because the interaction between these 2 proteins, SIAP and SIAQM, is really weak. And so we used microscale thermophoresis to test this. And we find that we get an estimated binding affinity of this protein SIAP for SIAQM of 400 micromolar or 4 millimolar. And the binding affinity of this value is considered very weak and transient. So that confirms also sort of why we don't see this interaction on a grid. And you can also use analytical ultracentrifugation to measure and monitor protein-to-protein interaction in solution. So we did this for the SIAP and the SIAQM, and we see that we get only about 3% complex formation. So consistent with the microscale thermophoresis, we see that also the interaction in the AUC is very weak and very transient, which may be why we can't capture it by cryo-EM. But also note that these experiments are done in vitro, and maybe it is actually that the transporter is sort of stuck in this downward conformation where the SIAP wouldn't be able to bind, because that's also what we see in our cryo-EM structure. So to probe and to understand complex formation in more detail, we took an alternative approach to trying to get a complex with EM. And to do this, we solved the structure of the SIAP to model it later in complex with the SIAQM. So we have these really beautiful crystals up here that diffracted to 1 angstrom by X-ray crystallography. And in our structure, we had sialic acid bound. And so this structure is for SIAP from Photobacterium profundum, which is the cognate SIAP for the SIAQM structure that we solved by cryo-EM. And so SIAP proteins are comprised of both alpha helices and beta strands, and they're described as having 2 domains, an N-terminal domain and a C-terminal domain. And so SIAPs undergo a really large open to closed transition that is often compared to or likened to that of a Venus flytrap. And to do this large opening and closing movement, they bend around these helix regions seen in the alpha helix and the beta strand here, so the hinge. And something else I want to point out on the slide very quickly is just these 2 caliper helices here, because I'll mention them later, alpha 2 and alpha 5, and also this beta 6 and beta 8, which I've labeled as an interaction hotspot. So as you can imagine, this surface here is the bottom of the SIAP. So you can imagine that that's actually what's docking to the SIAQM to deliver this sialic acid here. And so if we take a look at the sialic acid binding site in more detail in SIAP, the sialic acid is in this green or cyan color here. You can see that it's bound very tightly in the SIAP by these 2 twin arginine residues that form a salt bridge with the carboxylate group of the sialic acid. And there is also this hydrogen bond network over this side here that links the sialic acid from here all the way down to this R49 residue, and which is in close proximity to this asparagine 148 residue. And we describe this area down here, which is at the bottom of the SIAP, as a latch. And we think that this latch may sort of allosterically facilitate upon SciP docking, it may open up and then facilitate or disrupt this network interactions and allow the sialic acid to then be released and given to the trap for the SciQM for transport. In solving our structure at 1 angstrom, we could see that we also had a really nice ordered water or solvent network in the substrate binding site. And recent work has shown that this ordered solvent network is really important for SciP specificity. So if you mutate a residue outside this ordered water network that disrupts the network, even if it's not directly bound to the ligand, it will really reduce the specificity of the SciP. So we hypothesized that then upon docking of the SciP to the SciQM, there are surface interactions between both of these proteins that may disrupt both the protein and the solvent-mediated contacts with sialic acid to then allow the transitions required and the sialic acid to be given to the transporter for import. And so here we have a model of the trap complex based on coevolution. So we used our cryo-EM structure and our crystal structure of SciP. And we used 3 different programs, Gremlin, RaptorX, and AlphaFold, to make a model based on coevolution. We find that we have high coevolution signal, surface and charge complementarity, and also high sequence conservation observed between these 2 proteins. And these rods over here, the colored rods here, these indicate high coevolution signal. And also the top of the SciQM here kind of has like a bowl-like shape. And then if you flip the SciP over and you look at the bottom of that, you can see that it also has a complementary shape that would nicely fit over top of the SciQM. And an interesting point here is that these contacts you see here with the rods, they involve mainly surface residues of the scaffold domain, which is this sort of half of the transporter up here. The only interaction we see between the transport domain is in this residue, this region labeled with an A here. And so this is, you can imagine that this is really important for elevator movement, that the SciP is not actually docking to the transport domain, because the transport domain still actually has to move up and down. So this makes sense in that respect. So as I mentioned, we also have sequence conservation at the interacting surfaces. So between the SciP and the SciQM, we have clusters of conserved residues at the surface, and in particular on this C-terminal domain of SciP over here. And the sequence conservation in these figures is indicated by this pink or red color here. And you can see it's predominantly over this side. And then if you take a look at the top view of the SciQM here, you'll see that we have the complementary red color over here in the sequence conservation, which makes sense if the SciP docks to the SciQM in this position that we're predicting. And interestingly, we also see this sequence conservation at the interface between the Q subunit and the M subunit, which makes sense because, I mean, evolutionarily, these want to always stick together so that they'll still work. So it makes sense that we would see some sequence conservation in these areas. Cool, and if we take a look at the interacting surfaces between SciP and SciQM in a little bit more detail, we can see that there are some hotspots. And these hotspots are comprised of a bunch of different potential salt bridges that form. So on this side of SciP over here, this is the alpha-2 caliper that I mentioned earlier. We have this salt bridge that forms between arginine 49 and aspartate 50, and we also have one that forms between aspartic acid 59 and arginine 292. And on the other side of the transporter over here, so the C-terminal domain of the SciP, we also have these interaction hotspots of potential salt bridges. So we have this D181 that forms a salt bridge of K232, and we also have K143 that forms a salt bridge with D36. And in general, we think that these interaction hotspots and potential salt bridges are contributing to both the recognition and the allosteric modulation of CIP. So they are contributing to the recognition by recognizing where the CIP should bind and dock to the QM, and the allosteric modulation, it may be that the salt bridge networks sort of disrupt any of the ligand binding, and they allow the transitions to occur. So in particular, we think that this K143 to D36 salt bridge here, we think that because it's 2 residues away from arginine 145 that I mentioned earlier forms a salt bridge with the sialic acid, because it's only 2 residues upstream, you can imagine that when it docks to the SiQM, that it may sort of pull on this strand and then disrupt this bond, which would allow then the sialic acid to be released as the CIP opens and then given to the transporter for transport. Cool, and so here we have a model of the elevator transition, which I'll talk through a little bit. On the left-hand side here, we have the inward-facing elevated down structure. And so this is our CIP crystal structure and our SiQM cryo-EM structure. And we aligned these, this complex, docked and aligned it based on the complex that you get in alpha fold. And you can see as I have we have the elevated down conformation, we have the substrate binding site at the bottom of the transporter, or at the bottom of the elevator just here. And on the right-hand side, we have this outward-facing elevator up conformation. So again, here we have our CIP crystal structure, and here we actually have a composite structure of the SiQM. So being a composite, I mean that we have our cryo-EM structure for the scaffold domain over here, but we've made a homology model of the transport domain based on LA-Indy, which shares the same fold and topology in the M subunit, but the structure in LA-Indy was solved in the up conformation. So we have a homology model just for this portion here. And now we can see that our substrate binding site is sort of at the top here, in a really nice position for the CIP once it opens up to sort of drop that sialic acid in there for import. So you can kind of see from these figures that the transport domain moves down and it moves up through the membrane against this really rigid scaffold domain, which is just sort of holding the transporter in place there. And this allows, therefore, the substrate binding site to achieve alternating access and to be open on the outside and then open on the inside to then release the substrate for import. So we know from our structures, the SiP and the SiQM, that we have these really large conformational transitions for both the proteins. So for SiP in the pinky red color at the top here, we have this really large opening and closing transition, which can be likened to that of a Venus flytrap. And for the transporter at the bottom here, we have this really large up and down movement, so the elevator transition. And so we propose that, we know that these happen, these conformational transitions happen, but we don't know how they are linked. We propose that the substrate binding protein is operating the transporter, so it's sort of operating the elevator, but whether it is causing a global conformational transition, so is it binding and is it causing the transport domain to move up and down by pulling it up or pushing it down, or is it just causing local gating rearrangements? We're not too sure. So maybe it's just binding and it's causing some small rearrangements just to open the gate, and then the sialic acid can be shuttled in there. So we propose a new mechanism called the elevator with an operator mechanism of transport, and I'm going to take you through this in a little bit of detail. So this mechanism, in position 1 here, we see that the operator, which is the SiP, captures its passenger and takes it to the elevator. So this is CyP binding the sialic acid outside of the cell and then heading towards the transporter for import. And in position 2 here, we have the operator pushing the button of the elevator. So here we have the CyP binding to the CyQM, but whether this binding is causing this large conformational change and the pulling up of the elevator, we're not sure, or maybe it is already spontaneously up the top there, and the CyP just enables some local gating rearrangements. Number 3 here, the door to the elevator opens and the passengers are ready to be loaded. So the gates are opened on the CyQM, the sodium is able to bind, and the sialic acid can then be shuttled in from the CyP once that opens up. And then once the elevator is loaded with its passengers, the door is closed, so the gates are closed, and the elevator is sent back down to the bottom floor. So this is the transport domain moving from the top to the bottom. And then lastly, we have the door opening at the bottom of and the passengers are able to exit the elevator. So this is the gates opening on in the transporter at the bottom and allowing the substrates to move. And so ultimately, this is alternating access and how the sialic acid is imported into the cell. And so I'll just wrap up now. In summary, trap transporters are a unique family of secondary transporter that require a secreted substrate binding protein for transport in a manner analogous to ABC importers, but the ABC importers are obviously primary transporters. Traps are really interesting, being the only secondary transporter that we know of that uses this substrate binding protein to deliver substrate. Traps are only found in bacteria and archaea. They're not found in eukaryotes, not found in mammals or humans. So this opens avenues for new antimicrobials that may be non-toxic to humans. We solved the very first high-resolution structure of a trap transporter by cryo-EM. We solved this in both amphipole and in nanodiscs, and this allowed us to define them as elevator type transporters. And we found that our elevator transporter has an extended scaffold domain relative to others, and this likely enables them to operate as monomers as opposed to the typical oligomers you see for elevators. We found that substrate uptake requires the cognate substrate binding protein, and that transport is electrogenic, requiring at least 2 sodium ions to transport a negatively charged sialic acid across the membrane. And so our cryo-EM structure and crystal structure of the cognate substrate binding protein, together with co-evolution driven docking studies, provides a molecular basis for substrate import that we describe as an elevator with an operator mechanism. And lastly, a big thank you to the team. So this has been a really large and international collaboration over many years, involving many different people from all over the world. So first up, we have the team at the University of Canterbury in New Zealand in Ren Dobson's group, and especially James Davies and Michael Curry. So these guys, they started this project during their PhD, and then they continued on with it when I left UC. And we share authorship on a preprint that we have on bioRxiv, and we hope that this will be published soon. But if you like this talk and you're interested, then please feel free to take a look at that. Then also there's the team at Stockholm University and SciLife Lab, David, Drew, Ashu, and Dustin. We solved the cryo-EM structures together. And then there's Cesare and Maria Francesca at the University of Calabria in Italy. They did the proteoliposome substrate uptake assays for us. And then we have Peter and Sam from the University of Otago in Dunedin, and these guys helped us develop antibodies for our trap transporters. And we also have Jane and Jack from the University of Auckland. They've been involved in molecular dynamics simulations for this project. And then we have Rams, Rosie, and Soichi from different places all over the world. These they have been collaborators sort of since day one, have been involved in this project for a really long time. And then of course we have all the funding agencies at the bottom here that made this work possible. So thanks to everyone there, and a big thank you to all of you for listening. I hope you enjoyed it, and I would be happy to take any questions that you may have.
Wow, thanks so much, Rachel. That was an amazing talk. So much stuff in there, and it's been a super cool journey, and I think a really scientifically interesting endpoint. I'm sure lots of people have questions. I think the way this works is if you raise your hand, I can allow you to talk. This is the first time I've co-hosted a Zoom session. But while people figure that out, one thing I've always sort of couldn't get straight in my head or understand about proteoliposome reuptake assays is it possible for there to be different topologies within the reconstituted proteoliposome, or is there just such a positive charge on the cytoplasmic side of your transporter that it's just like strongly preferred?
So are you asking if there could be transporters this way and transporters this way?
Yeah. And then like measuring the reuptake, you would have to kind of somehow normalize for that.
I haven't done the proteoliposome assays myself, but I would assume that, of course, they would go in both directions. But for this system in particular, it's quite nice because you don't get uptake without the CIP there, and the CIP isn't going to be on the inside of the proteoliposome, so you're never going to measure transport going the other way. So we would only be measuring the transport going in anyway.
Oh, cool. These heaps. I think I see one hand up. Gil, please.
Unmute. Can you hear me? Sorry.
Yes, I can hear.
Thank you, Rachel. It was a terrific talk. I really like the elevator analogy. Very cool. I'm kind of curious. So you solved the structure in the elevator down conformation. And I'm curious, how do you then make the leap to sort of realize this is an elevator style transporter? Is that based on sort of similar structures of similar transporters where both sort of up and down structures have been solved, or is it just looking at it and realizing there's no other way it could actually sort of, take the substrate in? Like what's the sort of the,
we can kind of see by the structure that we have this scaffold domain and this transport domain, but we made that observation based on the topology it shares with the this other family of transporters, the ion transporter family. So this is what VCND is, and these are elevator transporters. So they share a really, really similar topology to them.
Right. Okay, cool. And is it something you're interested in doing, is solving sort of the elevator up sort of conformation just to validate everything, or is it like you
yes, for sure. We're absolutely trying to do that. It seems that, so we did like 3D variability analysis with our cryo-EM to make sure that we didn't have any alternative conformations, but it seems that our nanobody is really locking it in this elevator down conformation, which is really nice for the resolution that we got. But of course, we would really like to get the up conformation as well, so that we can actually model this transition and sort of model, then we can also better understand how the CIP is maybe even delivering the substrate and things like that. And we'll do that by, first of all, like we want to get a complex, and then hopefully a complex will be in this up conformation anyway. But also we have a bunch of different nanobodies that we can screen that might bind in different areas and sort of stabilize this other conformation.
Cool, that's very cool. Thank you very much.
Thank you.
Great. I can't see any more hands, which means I get to ask more questions,
okay.
Which is fine by me. So one thing that's always fascinated me about secondary active transporters is how the secondary substrate provides, energy for conformational transitions. And can you form a hypothesis about how the two sodiums kind of drive that elevator transition down its concentration gradient?
Yeah, kind of, but it's just a hypothesis. So we hypothesize that the two sodium ions actually bind first in the transporter. And we think this because they've actually seen this in the indie structures as well. They've done some assay where they found that that they don't get substrate binding without sodium present in their active site there, or substrate binding site there. So we think that the sodium sort of bind first in between these two broken helices, and this probably causes some like small local rearrangements and sort of rearranges the binding site for the sialic acid to bind.
Amazing. Thanks, Rachel. And I have another question, if no one has any. What struck me with the sort of substrate binding protein is really like how that mechanism of sequestering substrate seems to be unique to bacteria, and like why do you think that evolved, and why isn't it more common throughout evolution? If it's like obviously, you know, it provides a more efficient way for the bacteria to scavenge sialic acid in your case, but yeah, why is that kind of seems to be a unique solution?
Yeah, I mean, it's really cool that it's only found in bacteria and archaea. I don't really know why eukaryotes haven't evolved this, but I imagine that it has evolved to be able to scavenge very scarce nutrients. So the SIAP substrate binding protein has a really high substrate specificity. So if a nutrient is scarce, it's going to bind that with really high affinity and then deliver it to the transporter. And we think that our trap transporter has actually has quite a low affinity for sialic acid, because our binding site isn't very charged, and you would expect it to be quite charged to bind sialic acid tightly. And also we don't see the sialic acid in our structure. So yeah, we think that the trap QM component has really low affinity, and it probably wasn't able to scavenge the sialic acid in these scarce environments without the SIAP present. And I mean, some environments there's going to be a lot of sialic acid there, right? But usually these would have other sialic acid transporters like the sodium solute importer. But for us, the Photobacterium profundum is a bacteria that lives in the bottom of the ocean. So it lives in an aquatic environment, and you can imagine here that sialic acid is probably really scarce here.
Yeah, cool. Great. Well, unless anyone has any last questions, just remains for me to thank you so much once again, Rachel, for taking the time to share your amazing research with us.
Thank you so much for having me.
No worries. And yeah, go enjoy the summer day in Stockholm.
Yeah, thank you. Cool.